I'm an entomologist, which is the study of insects, but we pretty much get lumped into the study of all creepy crawlies. And in fact, on Maunakea, that's what I work on, is basically anything that's a little bit smaller than usually people observe. So I'm going to be talking about a lot of the research that I've been conducting on Maunakea and some of my students' work. And hopefully I can do this in a way that's factual and interesting and quick, so that we can have some questions, because I know there are lots of environmental questions on Maunakea. And there are many of us who actually work up there often, and perhaps we can actually have that discussion of what we've seen, what we recommend to Office of Maunakea Management and DNR, and basically whoever will listen. And it happens a lot. So there are many little things that help make Maunakea special. Of them, I get to study, and lucky enough to study many of the little insects on Maunakea. So who am I? Basically, I am an adventurer in the details. So I grew up in Pennsylvania, far away from here. Basically a free-range kid with my brothers and my mom and my dad. So we would just go out and look for things and admire and become interested in what there is out there. That led me to really explore it. My brothers and I, we would go to the library and get kicked out of the adult sections for checking out the human anatomy and physiology books. So that's who we are and where I came from. So through that, I went to college and enjoyed it and liked teaching and wanted to figure out how to continue to do teaching in a way that was of more interest to me. So I went to study insects. And luckily, I got to go to Oklahoma State and study squash bug immigration and emigration and damage on watermelons. But basically that was for 2 years during the summer, from dusk until dawn, catching insects and painting them with little colored dots and counting them the next day. So about 3 months of that for trying to figure out how far insects travel in a field and in the native environment. And then from that, I was like, this is fun. It's a little, my OCD comes out and I can count those bugs. And I wanted to figure out how to continue to do that and get a more broad training, because within insects, there's always going to be more to know. There's 20 million of them, all right? 1 million of them are described. It's about the same ratio, give or take, here in Hawaii. About 4,000, 5,000 described, 10,000 endemics. So we still are learning a lot about those creatures. I got to come to UH Manoa for my PhD in 2005, applied for the position like a job with my PI, Dan Rubinoff, to study the Wekiu bug at the summit of Maunakea. Within entomology circles, I had heard of this thing, this little creature that lived on the summit of a wonderful mountain on Hawaii Island, and got to study it from 2005 until 2012 when I finished. So I'll present some of that information here. But in so doing, I'm a country boy. I'm a mountain guy. I'm not a water guy. And I love this island as much as hopefully everyone else does who's here and interested. So what do? I'm a newly minted assistant professor of applied entomology at UH Hilo. I get to teach insect and animal-related courses in the College of Ag, Forestry and Natural Resource Management there at UH. And I am also graduate faculty through the master's program, so that I have some graduate students. And my job is to research arthropod-related topics as they apply to natural resources. So it's very, very broad. Part of my job is actually to work on Maunakea to understand that as part of the mission of the university. Here's some of my students and collaborators. This is what we do on Maunakea. We basically go to 9,000 feet, and as I grew up, through basically my parents are hippies, so Native American culture was part of my growing up too, and it was ask permission and give thanks. So that is how we go into the field. I think this is really essential. We have protocols for all the gear we bring, and we've actually had a debate in my lab whether or not we write on there, stop a moment on your checklist and ask permission and give thanks. Does that cheapen the actual act of asking permission? And we haven't come to a conclusion yet. It's not on the checklist yet, but we do it. And those are the kinds of things that I'm really interested in having more communication with, to meet and with people for that common ground. Insect conservation. This is a lot of the work I do, but conservation can be defined in a couple of different ways. Conservation is conserving resources. It's not necessarily preserving without touching. There are different ways to conserve resources through management, and that's what I do as an applied scientist. Insects are the bulk of biodiversity in any terrestrial system. They are all of it. In Hawaii, it's about 70% of all described things on lands are the insects. Actually, all the encountered things, not just described. And these creatures are the mediators of trophic level interactions. But what that really means is they're the little guys that are in the little things that you can never see. And they're moving energy, they're moving resources around, and they're killing things, they're killing each other, and they're eating everything else, and they're dying. And that's what their role is. They are the mediators. And they're also an overlooked metric. So getting into the science of this, how do we really understand and appreciate and then convey that these insects are a real part of the balance of life, okay? And this is where I work most of the time. It's because of the university system. The university has management aspects. Let's see here, I think I have a laser pointer too. Whoops. So I've done most of my research here in the management corridor and in the Maunakea Science Reserve, including the astronomy precinct at the summit of Maunakea. A lot of what I do now was based on recommendations that were approved in this Natural Resources Management Plan from 2009, including abundance and density of the insects, arthropods that occur in this area, the alpine stone desert. Also work on conservation strategy to preserve and protect those resources. And how to assess the risk that humans pose, because there are many of us, and we can travel much faster than we used to. We move things around. And how do we deal with that? Basically, in an area that is about as natural as you can get, given how people have moved and altered things. How do we deal with that? Part of that is this wonderful creature, the Wekiu bug. Here's an abridged history of all we know about this insect. Described in 1983. It was actually found by some entomologists and natural resources gurus back in the late 70s and early 80s, and was formally described in 1983. Little hiatus. This is usually what happens with entomology if it's not a pest. Someone decided to try to look at the insects at the summit of Mauna Kea and see how they survive winters. Okay? How do they go below freezing and survive? And they looked at the Wekiu bug and our Lycosa spider, a wolf spider that's at the summit, and a caterpillar, which we'll talk about those as well. And they found that, those insects are super cool. That's the joke. Everyone's heard it now. But they do super cool. They can go below freezing temperature and not freeze. The Wekiu bug can. They actually tested whether or not it's typical antifreeze glycol proteins, and it's not. It's probably something else, probably salt water. Just they desiccate, and they can survive the desiccation, and they're probably very salty, so they don't freeze. The caterpillar can freeze solid and then thaw out and cruise around. But there are a couple of different ways that can happen. And then they looked again in 1998 and found, I believe it was 7 Wekiu bugs. And that's what started these petitions to list this species as an endangered species, which of course, in our system, draws forth lots of actually money, because you can't do anything without money, at least in this aspect. So there was lots of research from the year 2000 until currently. It's ongoing. Some of this research that I'm going to present here was part of the Fish and Wildlife decision to not list the Wekiu bug as an endangered species. It's not an endangered species right now. It has all of the wildlife protections that all native insects have in Hawaii. It's actually quite a lot. You can't do work on native insects without permits. It's actually more so than any other state. It's actually pretty cool. To start off the bat, we'll go back to this at the end, but I did do work through the TMT site surveys long time ago, I believe in 2009 when I was out in the field, to look if there are Wekiu bugs in these areas where they were proposing to put the TMT at that point. And what we found, this was only one of our efforts. There were many other efforts throughout the years. But there are no Wekiu bugs regularly found in that area. We found a few over the years, but very, very rarely. Mostly what we find are Wekiu bugs in and on pu'u themselves, on the cinder cones. And there's some physiological reasons why. So this was a lot of the controversy, was there are Wekiu bugs occurring right next to that access road that they're intending to open. How do you deal with that? As a scientist who knows about insects, we can at least have that discussion. Are we going to destroy all of those insects or somehow mess up their life history of the entire species in this area in this way? And those are the discussions that we were having. Why all the attention? Other than the 30-meter telescope and Endangered Species Act, it's an incredible, unique Hawaiian thing. This is a Wekiu bug. I have a couple other macro pictures, but it's actually very unique as far as insects go. So it has that going for it. Of course, its reported rarity goes head-to-head with the astronomy development. And this is what caused all those studies to kind of ramp up. I don't know if my numbers are right or not. I just went 1.5 billion. I think I heard 1.4. But when you have that much money against any kind of environmental resource, there's going to be a controversy. It is how it works. And of course, not just that, but there are then state and federal requirements to protect native species. And there is, and there always will be, a little bit of resources to study these things. So here's a summit from Google Earth. Originally, with the Bishop Museum studies and from 1980 or so, Wekiu bugs were only known from the summit proper, okay? Pu'u, Haoki, Wekiu, that area. The Bishop Museum was contracted to then do some more research, and they found Wekiu bugs in these other pu'u, Mahoi, Poipoi, Maunakea, as well as down here in some unnamed cinder cones. This is Lilinoe right here. Down that far, about 11,200 feet, the Bishop Museum found Wekiu bugs. So they expanded the overall range. What they also found was Wekiu bugs are pretty much never found in that glacial till, the gray area between cinder cones. So that means something. How do we deal with that when we know about these Wekiu bugs, which are flightless? They're not going to fly between cinder cones. They have to walk like the rest of us. And how do they deal with it? So this is, from the summit, the habitat through which Wekiu bugs need to crawl and through which apparently they cannot survive, at least not through long term. They'll only be able to survive on pu'u. And we can see that there are impacts on those cinder cones, correct? Here's Subaru Kek. An area right here is where I've done lots of my Wekiu bug work, right down in Poibol. And these insects are found right next to impacted areas that are obviously disturbed. So when you look at insect work, that means that perhaps restoration is possible for this species. It's basically never works very well for other endangered species unless you've already seen that they can survive in these impacted areas. How do we catch them? Shrimp. Shrimp paste. Delicious in recipes, but it basically smells like rotting arthropods. Shrimp are related to insects, and these Wekiu bugs are scavengers. They'll eat anything that smells dead, okay? So we put pitfall traps right under the cinder with 2 cups with a little wick and some water, and we leave them out for about 4 days so that the bugs have food and water for that long. We catch and release, and we count them. I haven't painted them yet, maybe next year. Here's the little guy. This is actually a little girl. This is a female. These are the wings. They're flightless, micropterous. Makes them very unique within the groups that they're related to. They're scavengers. They're eating other insects. All of its cousins, all of its relatives, which are also endemic Hawaiian insects, are all plant feeders, seed feeders. So that adds to the uniqueness. And those are all criteria for endangered species listing, actually. And they only occur in this one area about above 11,000 feet on Maunakea. This is their food. This is snowpack, snowfall on Maunakea with insects that have fallen out of the sky and landed there and died. Wekiu bugs can't have no way to subdue prey. Whatever they eat has to die because of Maunakea, because of the environment up there. Snow, very cold, very dry. These insects will fall out of the sky and die there. Why do insects fall out of the sky at the summit? Insects don't have very good brains. Many insects are known as hilltoppers, and they're basically going to always climb up until that level is off, and then they're going to try to land. Basically, they're looking for mates or for food. And up on Maunakea, where there's no plants or water to have them help survive, and then fly on somewhere else, they die. And all of the insects on the summit of Maunakea use this aeolian debris, wind-borne debris, as a food source. Here's all the Wekiu bug's relatives. If you're interested in this, see me afterwards. Here's a group. They feed in family groups, and they don't ever eat each other. It's actually really neat. This is a large introduced tachinid fly, and this is a pair of Wekiu bugs and some nymphs, and they're feeding on that as they would in the field. This is actually done in captivity. They have sucking mouth parts, so there it is, sucking out the brains of that same fly. You can see it has straw-like mouth parts going in between the plates in the top of the head, and that's how they feed. But there wasn't a heck of a lot known. So when we started, for any work, you need to know the animal. You need to know about the thing you want to know more about, which means you have to observe it. You have to experience what it experiences from its point of view. Its point of view is about from 4 millimeters and maybe 2 millimeters tall. Okay, so it's a very small area. So what we did in captivity was brought these from Maunakea to Oahu, to our laboratory, which is actually I think it was an 8-month process to figure out how to do that. And basically we were treating these insects as if they were endangered because that listing could come down at any time. And if we ever had them in captivity, we had to basically keep track of them as we would have for Endangered Species Act. And what we managed to do was have them reproduce in captivity, observe them hatching out of the eggs, these iridescent gold membrane of the eggs, the corium, and they molted and reproduced through different life stages. And what we got from that was physiological research of how they survive and what temperatures they need. What is really cool is the air temperature on Maunakea, as recorded by the telescope astronomy facilities, is almost never above their minimum threshold. So they cannot survive in the normal air temperature of Maunakea. The only way they can grow was with solar input, okay? So it warms up the rocks, okay? So this is what that means. In captivity, we raised these insects at a whole bunch of different constant temperatures and watched how quickly they grew. Watched them molt their skin, watched them every day. It was about 3 years, 4 undergrads and myself. We all watched them every day, counted all of their molted skins, and created a model to say they die, or at least don't continue to grow at temperatures above 30 degrees Celsius. 11.3 degrees Celsius, they basically don't have enough energy for them to accrue nutrients. So where do those temperatures occur on Maunakea? They only occur at the surface of cinder cones on the rocks during sunny times. Where the ash layer is and where the glacial zone, where that glacial till was, the gray area on the map, basically the temperatures fluctuate too much or are too cold, and those insects can't actually go through all of their generation time because they can't grow, because they're either going to die or freeze, basically, every day. On cinder cones, they're basically temperatures modulated because of the rocks, the rock tephra. On cinder cones, it's still moving, and it's about 12 inches. So 12 inches is what the bugs need to be able to thermoregulate and survive those temperatures and reproduce. So what we did was, in fact, I lost my temperature loggers because it snowed. Literally, I'm allowed to do work for 5 days at a time on Maunakea. Otherwise, I'd have to have a big permit to have a permanent emplacement, which I didn't have. And in fact, I lost my temperature data logger because it snowed, and it took me 2 years to find it, and it still had data. But that means that I had 3.5 months worth of temperature data where I could say how insects grow. They need and could use the temperatures where they're found, and it took about 3.5 months for a generation to occur. It was actually really a problem for the species. Many people thought they were going to be very slow growing because they lived in a very cold environment, and it was going to take a year for a generation. But during the summer, they're just like all other insects that are related to them, about 3.5 months for a generation. And then we simulated the temperatures that they experience in that rock substrate and did all the statistics and found as long as they're breeding normally, the population doubles about every 15 days on Maunakea. As long as there are overlapping generations. Okay, that's a key aspect. We also found that, lo and behold, when you simulate their actual environments, they do really well in captivity, better than any constant temperature that we did. And this is actually borne out in just about every entomological research experiment. When you simulate fluctuating temperatures, the insects do better. Males live much longer than females, and this is really important for in the field. Females have hard work to do. They have to lay eggs, and basically it will kill them. The males, they don't have hard work to do, and they can just live a lot longer. So from all that physiology, we have a reason why Wekiu bugs live in these areas that I outlined as pu'u, and not in the glacial zones, because there's no thermal regulation that they can do in those areas because it's been flattened by glaciers 15,000 years ago. We also did genetics work, not to get bored in the details, but what we found is all of the insects at the summit have lots of genetic diversity, at least for them. It's actually almost the smallest amount of genetic diversity ever found in any species, but for them, seems to be going fine. But at these lower fringe habitats, it looks like the most common haplotype gets down there and survives. It's basically statistics. But what that means is the summit populations are probably the source of all the founders that can then survive occasionally at these lower cinder cones that are actually much warmer, even at 11,000 feet. Which brings back to this original story. Is the summit more important? Is the range more important? Whoops. In fact, it's both. There's probably immigration and emigration between all these areas, just very infrequently. What's really neat is there's also a very similar species on Mauna Loa. And what our genetics bears out is, at least this is supported, 15,000 years ago when the glaciers had covered Maunakea, that cold environment was in the saddle. And those two species separated from each other on those different cinder cones and have never bred again. But how rare are Wekiu bugs? This is probably what many of you are curious about. One Wekiu bug in 50 traps in 2005. Very rare. We actually don't know if Wekiu bugs were actually getting more common or we're getting better at finding them. Okay? That's part of the research of what we're doing. What was amazing was in 2013, we found the most Wekiu bugs ever found. So that's wonderful. Until you look the next year. Ta-da! We only found 52 Wekiu bugs in 88 traps. So it's an average of, I think, 0.19 Wekiu bugs per day and per trap. But for insects, this isn't actually very surprising. It's a very low number, and there were recommendations made for Office of Maunakea Management for any kind of movement of their habitat cinder. Don't do it until we know more, and they followed it. Okay? So those are the recommendations that we can have as scientists. Throughout the years, Wekiu bugs are found in higher numbers in different areas. It really depends on how the winter was, whether or not there was snowpack a given area. This was the details from that day, those days in 2013 where we found many, many bugs. Adults and nymphs were found in various numbers. And then the next year, lots of red X's and some occasional finds. I'm going to blow them up a little bit. And it was mostly adults at the summit with some reproduction occurring in Pu'uhaukea. We went back about a month later and found the opposite trend. Whoops, sorry about that. Wekiu bugs were breeding. We were finding mostly nymphs, so we know that they were actually reproducing, that there wasn't something that had happened that year that actually had stopped them from reproducing, which was going to be a bigger issue. But this happens to also be the year of the drought, when Lake Waiau was very, very low. So all of those things, of course, are related. And what we figured out was when we did our 15 days of population doubling, we were almost right on. That population doubled in the time predicted. So again, we're validating these things that we figured out in the lab so that we can say, if we're seeing this, if we're seeing those numbers growing, then even if they're low densities, at least they're doing well enough to be predicted. And what about this year? February. We barely ever found Wekiu bugs in winter, but this year we did. Of a higher number than we ever found last year. So an average of about 6 bugs per day. Just 2 weeks ago, we did our spring sampling, and basically those numbers tripled. All right, so population is growing. It's actually growing less quickly than we would have predicted, but we don't really know what kills them in the field. And there were those late winter storms, right? The blizzards in March. It probably killed lots of Wekiu bugs. So what we want to do is use this prediction as a real-time model if there's an impact, okay? If a telescope is, let's say decommissioning happens, you're going to actually destroy Wekiu bug habitat if you're going to move a telescope off. Hopefully they're going to restore it. I mean, those are the things that we're trying to create, is a recommendation. Whatever you define restoration as, we can use Wekiu bug habitat as a model of restoration. And we can predict whether or not it works, okay? So these are the things for applied science to get at those questions. How can we actually make these changes in a way that's informative and good? What I would really like to see, because I'm an agriculture guy, is what they do in basically every state except for ours, of pest species growth models. You put in your county, you say your pest and what you planted, it'll tell you when that pest is going to basically grow and destroy your crop. We could do the same thing for Wekiu bug numbers. We just have to find enough detail. The information is in the details, which is why we're working with the geography department. We were just up in here with Nathan, one of Dr. Ryan Peroy's students, and he was in the field with us. He's a geologist, geographer, and he had never seen the insects. So we gave him, the time in the field to show him that. And what he did was, with satellite imagery, again, partially that common ground, we never would have had this information to predict and accurately reflect what Wekiu bug habitat looks like if we didn't have LiDAR imaging and all that from space. So this is actually part of how we can connect all of that. I mean, it's still pretty ethereal science for me too, but there's some practical outputs that we use regularly. This is our first run at what is proper, good Wekiu bug habitat in dark red. Here's Maunakea. This is the astronomy precinct. This is actually that road to the 30-meter telescope site that is part of the controversy for Wekiu bug. We can actually see that in detail with this type of mapping. So this is Nathan and Dr. Peroy. What we're doing is a diverse program to have better recommendations. From that population growth modeling aspect, at least for this species, direct point mortality, killing bugs in an area, doesn't seem to be a major concern. It's the habitat integrity in general that is more of a concern. It's that cinder cone habitat is very important. Ecosystems change by new invasive species, new plants, new insects, new predators. Basically, we don't want the mountain to become complex. We want it to be as simple as it is, which is about 15 species of native arthropods and a couple of plants and lots of rocks. Okay? The Wekiu bug is no longer a candidate, but OMKM is actually, and the state, are basically treating it as a very special case of an insect, and there's lots of oversight. Take that as you will. That's the Wekiu bug, but they're not the only guys, so I'm going to hopefully finish up here in a few minutes. Here's everybody else. There are lots of other insects in the subalpine and alpine environment. Here's a lot of science. We go up there regularly and try to figure out what arthropods are where and on what plants and why. So you've met this guy, but here's a caterpillar. That's that caterpillar that could freeze solid. It lives down in the cinder below the rock tephra and crawls up from below and eats dead insects. I like the movie Tremors from the 90s. It's like that. And here's the adults. There's 2 species. They're new species. Anderson Prestes from Brazil was at UH Manoa. He's working on describing those species with me. There's a centipede, which may or may not be endemic. Basically, there's 1 centipede specialist, all right? I have to get its number. There's small spiders and large spiders, and we're still working on these species, but do the same things that we do for the Wekiu bug. What we do have information is the Wekiu bugs are restricted to cinder cones, whereas the rest of the species we find throughout the entire 12,000 acres. So it seems like, at least for what we know, the Wekiu bugs are the most restricted and probably the most fragile in that environment. But here we go. At the start of Office of Maunakea Managed Lands at Halepohaku, we've been surveying plants for the past few years. We've collected many arthropods. Basically, we have to kill things. We cannot know whether or not these are introduced or native species without looking at them under a scope. There's no other way to do it. But I have numbers on how many we're killing related to how many are actually there. So that's a debate that I actually am happy to have. So this is what we do. We train field taxonomists. It shouldn't just be me. I'm broadly trained. We can barely train the people in Hawaii that are here in Hawaii. So this is, as a professor, this is what I need to do, right? So we have 5 people that are all working on Maunakea, and they're all familiar with those arthropods. In case, I'm busy, all right? There are other people that can do this. And here's lots of the creatures. So we're looking at specificity on plants and where they're found. Flightless leafhoppers, many more flightless things on Maunakea. Another flightless leafhopper on Hinahina. Chenopodium is one of our major study plants. It's pretty dominant in the area where we're looking. This is one of Wekiu bug's relatives, and the Wekiu bug is happy to eat that if it flies up to the summit and dies. We have Hylaeus bees, our native bees, which look like wasps. One of the interesting things is there's holes in our Stenogeny microphylla, the native mint, chewed by honeybees. Honeybees are not pollinating this plant. They're nectar robbing. So we want our Hylaeus bees, maybe not our honeybees in that area. Fruit flies attacking these plants. This species is extinct, but we find it all the time. So you never find it unless you look. So we're trying to figure out how to get that biological note back to the Bishop Museum. They were the ones in '94, I believe, that listed that as extinct. But it's not found at Kilauea anymore. It's found on Maunakea. And we're looking at the introduced species and the insects associated with those. This is one of my students' work, Heather Stever. And what she's doing is looking at more detail at these plants and insects and creating a model of efficiency. We tromp around on the mountain a lot. We probably don't need to. And we're going to have scientific evidence that says, you know what? If you go look on these plants 36 times rather than 100 times, you're not going to get a better answer. Okay? So this is the impacts that we have on the mountain, and we can now say scientifically, you know what? We don't have to do that. We have a really good reason why to limit our impacts with scientific basis. So why we do this, offset those challenges. It's really hard to identify insects. If we get so many, it takes a long time to do. Weather and terrain is unforgiving on Maunakea. It's not safe. We have to work in pairs, definitely. We've got satellite phone and all that stuff. It guides field surveys, minimizes effort and cost with good information. The bad guys, the ants. Why are ants bad? We'll save the questions till the end, but they're ecosystem modifiers. There are no native Hawaiian ants. They are moving things around. They're gathering resources in one place. They eat everything, and they kill the native insects. Paul Kruszyniski showed this on Maui and on Big Island. Wherever there are ants, we lose about 40 to 50% of all of our native insects, and then pollination doesn't occur. Basically, ants are the worst. This one particularly is proven to survive above 10,000 feet on Haleakala. It is at Saddle Road. It is at the sheep station there at about 7,000 feet. Argentine ant. If it were to make it to above 9,000 feet, it would start to impact this relatively intact ecosystem. This is part of the reason why we always recommend for OMCAM to have very strict monitoring and sanitation rules for any of the activities on the mountain. It's because of this one. But we have no idea what all the other ants would do if they were to get there. So we basically treat every ant encountered as a worst case scenario because there's no going back. This is a problem once it starts. You can't put that genie back in the bottle, all right? It goes back to those point source impacts versus impacts that breed themselves, and ants are one of those. So my other student, Jordan Zarders, is working on trying to figure out what are the best ways to look for ants, how to identify them rapidly, and then do that threat assessment with Office of Maunakea Management. Look at all the telescope facilities. Maybe telescope facilities have these ants around them, the base stations in Waimea or Kona or Hilo. Hilo doesn't, because I work there and I know we don't have the Argentine ant right there. But we can actually alter inspections, greater or less, based on what the real risks are in real time. Conclusions, Office of Maunakea Management is actually really great to work with. I can complain a lot about other land managers here and throughout the United States. And maybe it's because there is trust. I know that when I talk to them, they'll actually listen to me and actually take those recommendations seriously because we have a good working relationship. I want to have that relationship with as many people as we can. It's tough to do. The greatest immediate threat are ants. It's also the biggest ecosystem threat. We want to keep that stone desert simple, and we want to have a balanced approach to all the work we do. And it's really hard to do that with a small crew in a very harsh place. Back to this, I think you saw this already. There's not a lot of Wekiu bugs around in that area, but those discussions about the 30-meter telescope are real about invasive species and about how to manage them, and manage them with good input and interaction. Because if people don't talk to each other, then we'll never know. And that's how problems can get bigger. Lots of people actually fund this work. A lot of it is actually Office of Maunakea Management and Astronomy. Just yesterday, I put in a big NSF grant as a faculty. Now I can actually apply for those things. And hopefully then we'll be able to do this modeling in real time. That's what I'm trying to do. But we'll see. All right. I'm going to keep working up there, keep working this OMCAM as much as I can, and hopefully get back into agriculture work, which is what I was hired for. And if there are questions, I know I took a long time. Maybe we can ask some questions.
I will share. I didn't say how will road construction for TMT impact gene flow between populations of Wekiu?
So
I'm saying
I'm open to pronouncing this correctly. Basically, a 6-year-old was the last. Oh, sorry. A 6-year-old was the last one to correct my pronunciation. Wekiu, I believe. So correct me if I'm ever wrong. But how could road construction affect the gene flow? That gene flow picture that we had, cinder cones are generally connected at the summit by areas that are actually wider than the width of a road. I have pictures of Wekiu bugs crossing roads. Okay, they do cross the road. What we have is information. It's about 1 kilometer space between a cinder cone and an area that they can't survive. And that, at least as far as our evidence is, and it's fairly strong. We can talk about it if anyone wants the details. It's about a kilometer distance would be necessary to break up gene flow within the Wekiu bugs.
Okay, thank you. A permit for the building does not stop a take. Can you please describe what a take is?
So take specifically refers to Endangered Species Act. And you can basically have take of an endangered species as known and defined, and you basically have to mitigate that take. Basically, it's killing endangered species. Wekiu bugs aren't endangered species right now, but yeah, if they became an endangered species, then all of this work, basically everything on Mauna Kea, driving through a parking lot would be take, and we know that, so therefore it would be in the government records. So basically all activities, be it walking through a trail, all of that would have evidence of take and would have to be mitigated. Whether or not that take is enough to cause the extinction of the species, I mean, that's what the Federal Endangered Species Act is without federal government support, that thing is likely to go extinct. That's the Endangered Species Act. We're not there yet. It could happen, and then we'd have to deal with it. But right now, we don't.
Thank you. What is the difference between special and sacred?
So without stepping too much out of my comfort zone, I find just about everything that I get to work with as both. I don't see the difference between special or sacred. Through my experience, I don't even think about it. Everything is connected, and that's just how it is. So I'm happy that I get to work with these things in these places that is incredible and amazing, and I hope that the intention of trying to preserve them and help them is what gets me through, and hopefully that's what I can impart to anyone I get to work with.
So I have two questions that are similar. Please describe the cultural protocol that you do in the field, and then you say you ask permission before any activity on the Mauna. If the answer that comes is no, then what do you do?
So our protocol, because I'm honestly not very good at memorizing just about anything other than scientific names, so I do not have a place-specific OLI that I regularly use when I go to the field. But what I do with my crew is we stop basically wherever we're going to get ready to go on Maunakea, which is usually we stop at 9,000 feet, face the mountain to where we're going, and I first ask all of my assistants if anyone has an OLI that they would like to give. And if none of my students have that, we do have handouts of OLI, but what I do is just silently ask my students that we all face where we're going to go, and we ask permission silently and give thanks and wait for the answer. And whether or not that's an answer of wind blowing, or basically what I'm doing is looking for an insect to fly by. That's how I do it. And if there was ever an answer of no, I'm trying to think if there's definitely been a time where I didn't go up. I wasn't, basically I would get up there and not feel well. Generally, I'm fine at high elevations, but there's been a few times when it didn't feel like I was going to be healthy there. And that for me was a signal not to go up.
Those are great questions and great answers. So thank you, Jesse. Dr. Eiben.